Good morning, dear participants. A very warm greetings from the School of Pharmaceutical Sciences, City University, Ludhiana. As you know, we are running the certificate course in pharmacovigilance, that is of a 15 weeks program in which multiple sessions will be held on the times. There will be the 4 minimum quiz in between, and the fun final assessment will be the on the last. The 75% of attendance is mandatory for each and every participant. I request every participant to mark their names and their organization or affiliation in the chat box of the YouTube channel. So as we know that we have previously studied about the pharmacovigilance. What do you mean by the pharmacovigilance? What are the sources? What are the process? What is the ADR? Types of ADRs like augmented, bizarre, chronic, disease, and all. Now, in today's session, we are going to start the topic the causality assessment in pharmacovigilance as per the World Health Organization UMC scale. It is a structured reference guide for harmonized assessment of adverse drug reaction prepared based on guidelines from the Pharmacovigilance Program of India, that is PVPI, that is be run by IPC Indian Pharmacopoeia Commission and SEDISCO. अगर हम बात करें कि कैजुअलिटी जो है वो कैसे असेस करें, जो भी हमारा एडीआर आ रहा है वो हमें कैसे पता लगेगा कि वो प्रोबेबल है, वो लाइकली है, या फिर वो क्रॉनिक है, या फिर वो अदर थिंग है। ठीक है, हमें उसका इफेक्ट पता है, नोन है, अननोन है। तो आज के दिन हम ये पढ़ेंगे कि आज का WHO वर्ल्ड हेल्थ ऑर्गेनाइजेशन यूएमसी स्केल. So in the next slide, the inherent challenges of the adverse drug reactions are, it means there are so many challenges regarding the ADRs. So most cases reports concern suspected reactions where certainly is exclusive. See, Luca specificity. First, if we talk about the Luca specificity, if we are having an ADR, is it a perfect ADR? From which drug it is? Is it is having a diagnostic criteria or a diagnosed test regarding the particular ADR? अगर हम बात करें कि अगर हमने कोई मेडिकेशन ली है, उसका कोई अनवांटेड रिएक्शन आ रहा है अपनी बॉडी में, ठीक है, क्या वो उसी ड्रग से आ रहा है? क्या वो उसके रिगार्डिंग हमने कोई टेस्ट करवाया है कि ये यही वही ड्रग लेने से हमें ये रिएक्शन हुआ है? तो फिर द सेकेंड स्टेप इज डायग्नोस्टिक वॉइड. डायग्नोस्टिक टेस्ट फॉर एरियाज आर यूजुअली एब्सेंट. अगर हम बात करें जितने भी हमारे एएमसी सेंटर्स में एडीआर रिपोर्ट हो रहे हैं, उनमें हम देखेंगे तो 50% और अप्रॉक्स 50%, आई कैन से दैट approx 50% of diagnostic tests are missed for the areas. And then there is a ethical constraints, that is a re-challenge is rarely ethically justified. It is done 99%, it is done by the doctor, physician, or a healthcare professional during the field work, whosoever is near about that patient. Then the reality gap, what gaps are there? In practice, few reactions are certain or unlikely. Most of fall somewhere in between into the zones of possible or probability. There are too many gaps. We are covering that gaps through the causality assessments. Moving from institution to harmonizations, there are the three goals of assessment: decrease disagreement, align accessories on a standardized logic, classify likelihood, systematically categorized the relationship probability, improve science, enhancing the scientific evaluation and educational value of the individual case reports. If we say from the bulk, we are synchronizing that areas. If we talk about there are multiple disease according to our anatomy, if we start from the brain to the foot, there are so many organs present in our body. For example, हेड, ईयर, नोज. अगर हम इसे बात करें कि हमारे पास जितने भी बॉडी के शरीर पार्ट्स हैं, उनके रिगार्डिंग कोई ना कोई हमें मल्टीपल डिजीज मिलती है. और कई बार क्या होता है कि कोमॉर्बिड कंडीशन भी मिलती है हमारे साथ। कई बार क्या होता है कि जब भी कोई कोमॉर्बिड कंडीशन आती है तो उसमें भी कोई ना कोई एडीआर आ जाता है। तो जरूरी नहीं है कि वो सिंगल ड्रग से हो, जरूरी नहीं है कि वो मल्टीपल ड्रग से हो, वो कुछ भी हो सकता है कि इधर वो सिंगल ड्रग से भी हो सकता है और मल्टीपल ड्रग से भी हो सकता है। If we talk about the goals, if we talk about the first goal, decrease disagreement, align accessories on a standardized logic. It means we have to align the things. We have to manage the long IPD files into a short. For example, in the next session, we will study about the how to write the case also. So in this, we will align the from patient information to the details of the drug, details of the adverse reaction, their casualty. We have to align in a short form. If we called about classify likelihood, we have to categorize the drugs in a similar way. Then the third point, improve science and enhancing the scientific evaluation. The scientific evaluations are done by the VG-Floor or PVPI. The experts are sitting on the seats. They are doing regularly working on the system. So in this, while on system procedures, a precise quantitative likelihood standardized procedures are essential for routine pharmacovigilance. It means we are having so much works a day. We have to regularly sit. We have to regularly monetize each and everything very carefully in the system. The next things, what we can do, what not to be do. That may be distinguish valid from invalid cases, prove the connections between the drug or event, change uncertainly into the certainly, the decreases of disagreements between the accessories, classify relationships like likelihood, improve scientific evaluations. If we talk about what things are necessary to do, for example, uh, I am having a headache. It might be any reason. It might be of a stress. It might be of a screen time. It might be of having a reading in a low light or the light reflections, or it might be having some thinking power in our brain. I am taking the medicines from the market like Dolo or the paracetamol with acylofenac or diclofenac. They causes me the reactions. If I generally talk about the paracetamol is causing a hepatotoxicity. But for the regular use, if we are using paracetamol daily for your small pains or small fevers, then it might be having a later reactions towards your body. It means it will cause hepatotoxicity in your body and your liver will become fatty. The next is the WHO-UMC methodology. What is the methodology behind this skill? We have to develop in consultation with the national centers participating in the program for international drug monitoring. If we talk about the first step, there might be the clinical pharmacological aspects, case histories, drug properties, and the second step is documentation quality, reliability, and completeness of the observation. The methodology starts from our study. If we talk about we are a healthcare student, we are studying the science from our childhood, from 4 5 class, 7 8 10th. If we talk about the 10 till 10th science, that is the basic science. Then we go for the 11th and 12th for the biology, physics, chemistry, or math. That is the some part of the advancement. Then we go for the professional courses like MBBS, pharmacy, PharmD, or pharmacy practice, pharmacology, clinical pharmacology, or the pharmacovigilance. There might be the some books. I will talk you about the here is the one reference of the book. If we talk about the Bible of a pharmacology, that is the Goodman Gilman. If we talk about the uh, standard book of the pharmacotherapeutics, that is the Roger Walker, in which you can see there might be the case histories, some small theoretical case histories, because our adverse drug reactions are started from the years of back like 1980s or 1960s. Then the quality documentation. The quality documentation is the pre-reviewed journals also provide the quality documents. The books, standard books, reference books, they also provide the quality documents. You have to combine both the things in your practical field. For example, anybody is working in a hospital, they will see what the physical symptoms or the variations are coming into the body. They will analyze that, and they will go through the case histories as the previously literatured in the files. They will combine, and then they will become for the practical tool of our case reports. Then the system provides a method to assess reports in the absence of definitive diagnostic proof. If we are not having any diagnostic proof, for example, some ADR we are having, हमारे पास ADR तो है बट उसका कोई प्रूफ नहीं है कि वो एग्जैक्ट यही है, तो फिर उस केस में क्या करते हैं, हम अपनी जितना भी लिटरेचर हुआ था, आयुर्वेदा, सिद्धा, यूनानी, pharmacology, pharmacotherapeutics, or any other books that are standard books or reference books, IPC books, Indian Pharmacopoeia Commission, right? We will go for that. Then only we can go for the definite diagnostic proof according to the symptoms and reactions towards the body. In the next slide, now we'll talk about the different parts of the WHO-UMC scale. First is certain. In this, the reach challenges are satisfactory and necessary. If we talk about the time relationship, that is possible time relationship to the drug intake. Alternative explanation cannot be explained by disease or other drug. D challenges. See, in this only the important is re-challenge and satisfactory if necessary. For example, I am having a itching after eating any antibiotic. The doctor will prescribe me any anti-allergic reacting drug, and the stop the antibiotics instantly if we are having any reaction. That means when the antibiotic is prescribed to injected in our body, the certain reaction comes of itching. अगर हम बात करें हिंदी में, तो कि हमें जैसे ही ड्रग दी गई हमारी बॉडी में इंस्टेंट रिएक्शन आ जाना, तो वो क्या होता है, सर्टेन एडीआर्स होते हैं, एकदम से आ जाते हैं, बट उनका रीचैलेंज पॉसिबल होता है कई बार. The next slide, we will talk about the probable or likely. It is having a time gap. Means कुछ टाइम इसमें आता है आफ्टर ड्रग टेकिंग. अगर हम बात करें कि हमने अभी ड्रग ली बट अभी रिएक्शन नहीं आ रहा हमें, और इस ड्रग का रिएक्शन कई बार क्या होता है कि 24 आवर्स बाद आता है, 12 आवर्स बाद आता है, तो उसमें हो सकता है कि केसेस पॉसिबल है. फिर their alternative explanations is unlikely to attribute to disease or the other drug. The D challenges are response to withdrawal in clinical responsible. Nature of event. Definite pharmacological or phenomenologic specific disorder. Re-challenge is not required. Insights. The lack of a mandatory D-challenge is the primary operational difference between certain or probable. If we talk about the probable, it might be the adverse reaction that might be recovered, or the body might be recovered from the adverse drug reaction, that the drugs comes under the probable category. If we talk about the next, that is a possible. Possible means हो सकता है, नहीं भी हो सकता. देखा जाए तो एक पर्टिकुलर टाइम मिलता है दोनों के बीच में, ठीक है. मतलब कोई भी ड्रग लेने से हम कह सकते हैं ड्रग ड्रग इंटरेक्शन भी हो सकता है और नहीं भी हो सकता. तो कोई भी अगर हम बात करें, जस्ट वेट वन सेकंड. तो अगर हम बात करें कोई भी हमें वर्क करना है, फॉर एग्जांपल हम ले लेते हैं कि मुझे आज की डेट में 8 या 9 पेपर भेजने, पॉसिबल है, हाँ हो सकते हैं, ये हमारे ऊपर डिपेंड है. ऐसे ही हमारी जो बॉडी है वो ड्रग का रिस्पॉन्स देखती है ना. तो फार्माकोलॉजी में तो हम फार्माकोकाइनेटिक्स एंड डायनेमिक्स पैरामीटर पढ़ते हैं, बट If we talk about any other clinical practices, the patient is to be monitored regularly in the IPD. But if we talk about the OPD, we cannot monitor that patient regularly. But the time visits can monitor that patient's health regularly. The next slide, the distinction between probable and possible. The variables are alternative causes like unlikely to attribute to disease or other drug, and impossible we can say could be explained by disease or other drug. प्रॉब्लम में ये है कि हम कई बार उसको नहीं समझा सकते डिजीज या ड्रग की वजह से, बट जब पॉसिबल हो हमारे पास एडीआर है, उसका प्रॉपर डायग्नोस्टिक टेस्ट है, तो हम उसको एक्सप्लेन कर सकते हैं अदर डिजीज या ड्रग्स के थ्रू. दी challenges for withdrawal: response is clinically responsible in the probable, and in possible information is lacking or unclear. Probable requires a clear link to the drug and a stronger argument against alternative causes than the possible. Then is unlikely. There is no relationship, no improbable things are there, but not impossible. Time to drug intake makes a relationship improbable, but not impossible. Disease or drug provide possible explanations. The key distinction is the timing does not fit the pharmacological profile and the better explanation exists. They are very less in the signal strength. If we talk about the signal strength, see, there is a 100% signal strength. Sorry for the network issues. Then, if we talk about the signaling strength, see, the certain casualty assessment is having 100% of a signal strength because it can be happens during the IPD times. The patient is inpatient departments. The person is there. The doctors are prescribing drug. He is taking drug in front of the nursing staff, pharmacy, clinical pharmacist, any interns, they can detect instantly. If we talk about the signal strength of probable or likely, it is of 75%. The 75% chances to be exact, probable, or a likely condition. In the possible, it is a 50-50 chances. If we talk about any situations, they are having the both the ways. There are 50% is towards the positive and 50% is towards the negative relationship. Then if we talk about the signal strength of likely, it is having a very lesser chance, that is a 15% chances of having a unlikely conditions of the casualty assessments. Then if we talk about the insufficient data categories, there are conditionally unclassified, there are temporary status, they might be the event or laboratory test abnormality reports. See, sometimes if we go for the laboratory test, there are so many diagnostic laboratories in the India. They are having different instruments for the calculating the values. There might be some variations. Either if we are doing a study also, there will be the variations of 5 to 10%. If we are planning some research work also, uh, we are taking the 95% of the confidence interval and the 5% of the error. In the second part, unaccessible or unclassifiable, the report suggests an adverse reaction but cannot be judged because information is insufficient or contradictory. Data cannot be supplemented or verified. These can be permanent status. See, if we are having a adverse reaction, we are having it's no exact criteria why it is happening, or why the laboratory values are not coming sufficient towards that. We cannot provide the exact classification of that drug. Now, there is a difference between all the categories like certain, probable, possible, unlikely, in between the time relationship, alternative, D challenges and re challenges. If we talk about the certain, there is a plausible time relationship, and having a and in probable category there is a reasonable. Possible is also having a reasonable time relationship, and in unlikely there is a improbable time relationship. See, there is a reasonable time relationship between the two categories only, that is a probable and a possible. If we talk about the certain, there is a plausible. Plausible means we can pause that reaction on the spot also. But in likely, there is an improbable, but we can manage that adverse drug reaction. If we talk about the alternative explanations, there is a no other explanation in the certain category. There is unlikely disease explanation in the probable, and it could be a disease in possible, and in unlikely, it would be a disease plausible explanations. D challenges, they might be certain, might be stopped, reasonable in probable, lacking or unclear might be in the possible, but in unlikely we are not having any de-challenge or re-challenge. We talk about the re-challenge, there is a satisfactory re-challenge in certain, sometimes in probable we can have satisfactory and not required. In possible we can manage that reaction, or we can reduce that reaction by giving the extra drugs. For the last, enhancing safety through harmonization, while causality assessment cannot change uncertainty to certainty, the constituent applications of World Health UMC scale enables better signal detection and clear scientific evaluations. If we talk about that scale can change this certain uncertainty to certainty, no, it can only provide the information that this adverse drug reaction is coming in this category. Okay, apply these criteria to ensure valid harmonized data within the Pharmacovigilance program of India. And it is the guidelines are provided by the Indian Pharmacopoeia Commission and many more. If we go through the each and every part of the today's session, we have discussed only the one scale. There are another scales also like Naranjo, World Health Organization scales. We will discuss that one by one. Now I would like to go for the one quick revision. What is the causality assessment, and what are the parts of World Health Organization UMC scale? In quick revision, we will go for that it is prepared by the IPC in the PVPI program, and structured for the adverse drug assessment of adverse drug reaction. The challenges might be the lack of specificity, diagnostic void, ethical constraints. There might be the adverse reactions are really specific to the drug. Diagnostic tests for the ADR are usually absent. Re-challenge is ready ethically justified. The second part, from the bulk to we are going to the harmonize. If we are having a number of areas towards India, throughout the India, we have to manage in a single table form. We have to calculate the drug so that we can take necessary actions towards the market, withdrawal of a drug, or stopping of a drug, banning of a drug, or any, we can say, the alerts to be given to the society. The do and don'ts. After that, the methodology. The methodology is the basic part of the drug clinical histories, books, literature, clinical practices that are combined for the practical tool for the case. And the next, there is a certain strength. The certain strength is signaling strength is 100%. in which re-challenges are satisfactory and necessary. We talk about the probable. The signal strength is 75% and the re-challenges might not be required. We talk about the possible. And there is the signal strength might be the 50%, it might be possible or it might be not possible. In this, there is a one important point, the gray zone is available. The shift from unlikely to disease and to could be disease means unlikely to disease or could be a disease. They're having situations like 50-50, हो भी सकती है, नहीं भी हो सकती. तो इसमें डी challenges भी होते हैं, but they can manage only the long term diseases. After that, there is an unlikely, that is a 15% of strength, signaling strength, insufficient data, and this is the difference. If we talk about the each and every causality assessment in the nowadays also, we are having most of the cases from the probable category and the possible category. Only few are from the unlikely and the certain category. If we talk about the reportings in the AMC centers, while I have also worked as a deputy coordinator in the AMC center in the previous eras, that we have seen that in a particular area there are so many cases. If I talk about the Malwa belt or the Malwa region of a Punjab, there are so many accidental cases, there are so many alcoholic liver cases, there are so many patients of diabetes, there are so many patients of hypertension, chronic kidney diseases or acute kidney diseases, cancer. So they are having some way possible of the 50-50% or 25-30% of the ADRs. From that population, the 25% of population is having the ADRs in which we can say the 45% is of a probable, 25 is of a possible that covers the 70%, and the 20% is of certain, and 10% is of unlikely ADRs that are possible in the market. So this is all about the today's session. So uh, we have done the causality assessment as per the World Health Organization UMC scale. Once again, I must repeat the things that every participant is having a necessary of 75% attendance, then only the certificate will be issued to the participants. Dear participants, please take care of their attendance. The 75% criteria is mandatory. 4 minor quizzes are there that are also mandatory, and one final assessment will be there on the last of the course. This course will go for the 15 weeks with the different sessions. And because we are conducting only on the Saturday, that's why it goes for the 15 weeks. Otherwise, it will be take the more than 1 month or 1 month on. So a very warm greetings from the School of Pharmaceutical Sciences. Thank you everybody. Is there any query? So I would like to ask my team members. There is any query regarding the today's session? Yes, sir. I would like to ask the team members, Dr. Rohit, is there any query regarding today's session?
Sir, I have seen the comments and there is no query of the students, so we can proceed further.
Okay, thank you, Dr. Rohit. Thank you, everybody. Soon we will meet on the next Saturday. Today is only one session. Thank you. Thank you, everybody.